Most people did. Some people did. Like the last question in the exam said that you were going to do. That you were going to look at a couple documents that were posted online. This direction thing hadn't gotten posted, I think, till Friday, so I was late getting that up. But I did eventually get it up. And you might look at it and be, well, that looks really thin and not very clear, and yeah, I agree. So we're just going to work within the realm that we've got, and that realm happens to be my crumminess, so I'm not going to take out my crumminess on you. But you still have to go through and do it, because we have points assigned to it. So, really I want kind of two things. Ideally, you've already gone through and done some kind of initial processing of the two journals. They were both kind of beefy. In theory, everybody's gone through and read them. Yeah? Anybody go, oh, that was easy? No? Okay, just curious. So you would read through them, and the primary focus of this is not trying to understand explicitly what is stated within the journals, though in theory I tried to pick journals or journal articles that you could at least approach some of the material with. So we're looking largely about intermolecular forces and protein folding, which was what we had just talked about. So it should have been at least in a ballpark of your understanding ability. And what I wanted you to do was identify the important sections within the article. Okay? Introductions, conclusions, what else might we find in an article? Results, methods. Anybody find anything else?
Experiments.
Okay. What the actual experiments were, which kind of gets at the methods and the results. Maybe a discussion of those results. Okay. An abstract, something to lead you into the paper. Okay. So the idea was to hopefully find those sections, or at least to be able to distinguish, hey, this is a new section. They didn't call it anything, but this is clearly something different than what was presented before. And the idea is to be looking at the context of how the language is used to help clue you in that the language has changed, that we've had a shift in what's being presented. Does that kind of make sense? What this should sound an awful lot alike would be, I don't know, a literature review in an English class where they're asking you to write something. Where you're looking at what was said and why what they said means something. Okay? That's what we're trying to get at here. Except instead of doing it with a literature thing where our overarching idea would be some grand theme to the piece of literature, we're instead looking at what is the goal of that experiment. What were they trying to do? Did they actually set out to do it appropriately? Did they prove it right? All of that kind of fun stuff. So you're doing the same things that you would do for a literature review, except you are now doing it for a scientific article. Okay? I would bet you had a little bit of trouble with that, but I could be wrong. Maybe you all thought it was easy. But what I want to do for at least a significant portion of the day is to discuss those aspects and look at some of those articles or parts of those articles and try and address what things allowed us to clue into those pieces. Okay? Why would I have you go through and do this? Well, your final project is a writing assignment. Okay? That writing assignment should be structured and organized as if we were to go through and publish it. Okay? So it should have some organization and structure behind it. The closer it matches real-world examples, the better off we all are. Kind of make sense? What I have found in past classes is that you've had enough experience with reading any kind of scientific or pre-health is not the appropriate term, but kind of research articles that you're already pretty adept at it at this point. Okay? And I haven't really seen any problems within the writing at the end. Okay? So after the discussion, I want you to go back and kind of re-review the content and try and organize, effectively refocus back to those questions and try and elaborate on, reference the text, where did different things jump out at you, We'll be talking about some of those things today, so if you take good notes, that should help you put that together. This then is the initial writing sample that I can then give to the writing certified people and say, hey, this is what we did as the initial sample, and then when you guys turn in your final report at the end, that'll be the final report that I show them.
So when is this part due?
That's a good question. I didn't even think of that. When should it be due? Let's try that. When do you want it due? Don't tell me the end of the semester, that won't work. I mean, next week? Before spring break? After spring break? I think right around spring break makes the most sense to me. It's a little bit far out. Ideally, this would be next week, but I'm okay pushing it into the spring break range. What do you guys think?
Right after?
Right after spring break? Okay. So we can make it due the Tuesday after spring break.
We need to answer these questions, but do we have to do it in like bullet point or like go through paragraphs?
You need to do it paragraphs.
Okay.
The idea is to prove that you know how to write. And bullet points don't do that very well. So that's the Tuesday after spring break, which would be calendar.
21st.
That looks right. Anybody want to challenge that? No, because that's right. March 21st. Just a sec. Let me write that down. March 21st. What I would recommend you look at this at, well, it's got to get recorded anyway. What you look at this as is effectively a free 50 points. Okay? As long as you've reasonably put together some paragraphs, some coherent thoughts, it should be a free 50 points.
Then online or in person?
I'm fine with either, though in person's probably going to be a little bit easier.
Okay.
If you don't have a printer or don't want to pay to print, that's fine. You can just turn it online. Anna?
So I guess I'm a little confused. So are we writing a paper about the questions, or are we writing like our own paper?
You're writing a paper focused on those questions. So you're not trying to summarize the article, okay? And this is something that I tried in a different class, and that's what nearly everybody did. Was say, oh, this is what the article was saying about this, and I don't care about what the article's actually saying.
Okay.
I want to know how it is saying it, okay? Which is challenging. That's why we'll talk about it today.
Okay.
Okay, so we'll start to look at some of the things that would be more obvious than others and how those sections are differentiated. Okay? And depending on how many sections that you're marking up or talking about, I would probably break your final paper up into that many paragraphs. So you could say, well, the introduction presents topics in this way, and that is different from other sections because this is how it's formatted.
So it needs to be both articles, or do we choose one?
I would do it as a summary of kind of both, because there are different parts in both. So one of your first paragraphs, which is one of the things we'll talk about, is that these two articles are actually very, very, very different. Okay? One of them is one that you should be mimicking for your final report. The other one you shouldn't mimic at all, okay, as the broader scheme of it, because they have very different purposes and goals behind why they were written. And that's partly one of the things that we need to be able to decipher when we look at it. Does that make sense? Yes. So questions about that? Okay. I will make an assignment for this with that due date, so it'll pop up in Canvas saying that that's what's going on. I'll even put it in as an online submission if you want, so you could online submit it to Canvas, or you can give it to me by paper. Either's fine. Other questions on that one? Okay, so I'm going to attempt to close that. Hey, that worked. Which gets us to the final writing assignment / presentation. In theory, this is posted to Canvas. The only thing that might be missing is this last section, or those things that are highlighted. So as part of our writing assignment, you need to potentially work with someone to put together a presentation for the class about kind of genetic information. So ultimately DNA, RNA, that kind of material. And there are some exceptions to that which we'll address. And the reason being is that it's typically not really evaluated in any detail in a biochemistry class because that's typically reserved for a biology class. The amount of chemistry that goes into that is so kind of involved and in depth that it just typically doesn't make it to a biochem class, right? Because it can be very, very, the word I like to use is a rabbit hole. Okay? In your process of researching, you may very well fall down a rabbit hole, right? And just snowball after something, be like, oh, well, this sounds really interesting, I'm going to keep looking at this, and find out that you're now miles away from the topic that you were originally starting at. Okay? So that's going to be something that you'll kind of have to watch out for. This is why you can coordinate with me and talk with me about your assignment as you're going through and doing it, and I will try to rein you in so that you don't get really, really, really involved. Okay? So we've got several topics up at the top. The idea would be that we work in pairs, which then shortens it, and I don't need a whole bunch of different topics. If you don't want to work with somebody, that's fine. You can work individually. That's kind of up to you. So the goals behind this, there's twofold. One, it's getting the information down, and then one, I'm also having you reflect on and look at the scientists that put this information together. Right? And that's largely because the scientists kind of get ignored, or they get put on a pedestal for being these really brilliant, awesome people, and in a lot of cases they aren't. Okay? They're normal people that happen to stumble across a discovery, or just happen to get there first, and then in a class they get put on this awesome, oh, look at how brilliant and awesome they are, and really they're just regular people that happen to be in the right place at the right time. Right? And that's part of the assignment. I want you to be able to see that these people weren't just naturally brilliant in most cases. They just worked really hard to get where they wanted to be. Okay? And they just kept being persistent at that study. Okay? So it's to make the people that put the science together a little bit more real, and then it's also a chance to let you look at the science in a little bit more depth than we could ever look at in the class. Kind of make sense? So within the genetic material, I've broken it down into a couple different sections. We've got what's known as the DNA dogma, right, which comes back to Avery, Hershey, and Chase. DNA dogma is a very centralized term that means very particular things. That meaning was largely developed through studies done by Avery, Hershey, and Chase. Okay, so you would be looking at DNA dogma as a topic, and you could then look at Avery, Hershey, and or Chase. Ideally it's an or. Okay? And you would look at one of those scientists and say, okay, what was their life like? What did they do? And that's building into a brief biography. Same kind of idea for DNA structure, replication, transcription, recombinant DNA, which I'm actually interested to hear more about. And then the specifics of gene expression, also known as our operons, and we've got a bunch of kind of scientist names associated with that. Okay? If you look at this and you're like, I actually have a specific interest in something else that I think fits within the scope of the class, then talk to me. We'll talk about it, and maybe I'll go, yeah, that is within the scope of the class. That doesn't really fit into the boxes that I want with DNA, but that's within the scope of the class. Fair game, go for it. Okay? And that's what's happened with this last one and or two. It's looking specifically at KER channels, NMDA and AMPA receptors, and then potentially also, or as a secondary topic, the exploration of polyamine homeostasis. And for those of you going, why are you reading that off the slide? I know nothing about those. Nothing. Okay? But I can at least acknowledge that both of those topics are within the realm of this class, I think that's fair game. Okay, kind of makes sense? So, the other topics, other scientists are fine, you just need to talk to me. Ideally, you talk to me sooner rather than later, because what I will do is then send these topics, and I've already sent this list, to our librarian, and then next Thursday, I think it's next Thursday, we will meet in the library instead of coming here, and the librarian will show you what we have access to at Chandler Gilbert, okay, for different research materials, and can potentially set you up with biography information and getting to the other aspects of peer-reviewed journals and all that kind of fun stuff. Kind of makes sense? The larger parts of the assignment, pre-reflection, okay, this is kind of more for you than it is for me, but it's at least getting you to put it down. I want you to reflect on what you know about genetic material, okay, or maybe what you don't know. Maybe you have no concept of what DNA is. Okay, that's fine. Write that down. You're just trying to get that information onto paper so that you can go back and reflect on it at the end and say, oh, wow, I've actually really learned something through this assignment, or I learned absolutely nothing. Everything was a complete review for me, in which case you probably should have worked a little bit harder on the assignment. Okay? Kind of makes sense? Now we get into the actual assignment. We've got a biography. So I want a brief, what do I have, 1 to 2 pages. This is all expected to be double spaced, just so you know. Oh, I don't have a mouse, that's right. Frick. Okay. Nope. I want to scroll. Anybody know how to get my mouse back? Oh, I don't have a screen. Oh, let's try this. No. There we go. Look at that, I can scroll. I don't even have to see it. So, your pre-reflection, the biography, build a brief biography. This is again trying to look at the scientist. Okay? I said I was particularly interested in the recombinant DNA. And there's only one name associated with that one. It's Carey Mullis. The reason I'm interested in that is it turns out that he is potentially a little bit crazy, so he could be an interesting one to do a biography on. But he is like the one famous person from my wife's hometown. And my father-in-law just sent an email, hey, anybody heard of Carey Mullis? So, and then, yeah, they proceeded to mention all the weird things he's done. He's kind of like scatterbrained, which could explain the town. Yeah, so I'm interested to see that. If you don't want to do it, that's fine. But as far as the biography goes, his is probably going to be interesting. Like they were talking about seeing space raccoons. Right? So yeah, that could be interesting. The original research, his research is probably why we have the Human Genome Project. Without his research in recombinant DNA, we don't have the human genome. Okay? So his research is pretty up there. So what you'll have to do is find an original peer-reviewed article and looking at that research and evaluating it. Okay? Evaluating that article, pretty much doing kind of what you're doing for the assignment that we will be talking about later today, the one that's due in March. Looking at what the different sections are, how did they design the experiment, what was the result of the experiment, what does that mean for the broader scientific community, that kind of deal. Which gets us into the next part, the broader context. These people were known for something. What is that something? Evaluate that something, allow us to understand what that meant to our broader scientific community. Okay? Current research, see what they're doing now. Okay? That could again be a weird one for Carey Mullis because he's looking at space raccoons. Okay? Okay? Doesn't have to be current research that is in biochemistry. It could very well be that you find somebody is so scattered that they're out doing weird stuff. If they're dead, okay, look at what projects have kind of come from their research. Okay? And by doing some reading on these people, you will be able to find that information. And then your reflection, okay? Reread your before you start. So once you put everything together, you'll reread what you wrote at the pre-reflection and go, oh, huh, I have changed, okay, or my perceptions have changed. Some people have had some pretty massive epiphanies in how different their thought process was, because they came in hearing one thing about a scientist and then did some research to find out that it was completely not true, that their perceptions were way off. Right? So it can be kind of enlightening in that respect. The last thing of the writing thing, which should be kind of before the reflection, is an annotated bibliography, just to show that you've looked up peer-reviewed articles and that you know what they were talking about briefly. Okay. That the librarian will go into a little bit more detail with, because we're referencing the ACS style guide. That is a link that will take you out to how you should format a bibliography. Okay. And with freaking technology today, you don't have to write one. You can just go to your paper and just be like, give me a bibliography, and it spits it out. It's irritating. The last part is the presentation. Okay? So within the writing assignment, even if you're working in a group of 2, you will still submit 2 unique reports. Okay? An easy way to do that is if you picked that first one, DNA Dogma, well, you had Avery, Hershey, and Chase. One of you can talk about Chase, one of you can talk about Hershey. Okay? Easy way to differentiate the assignment. Okay? You can still work together on finding content. The best way that I would present how you work together on a writing assignment, okay, in a group situation, if you're supposed to provide unique assignments, is you can talk to each other, but you can't see each other's document. As long as you never lay eyes on their writing assignment, you probably will not be plagiarizing. Okay? And that's the biggest issue with those group assignments, okay, or the group research. Make sense? Okay. Last part is the presentation. You'll then summarize everything, put it into a presentation. The writing assignment does have a due date. I don't remember exactly when it is, but I think it's a couple weeks before the presentations actually start. So that's to give me time to really quickly scan through them and be like, this stuff is really cool. I think you should talk about this in the presentation. This stuff is really cool, but this is so far outside of the realm of what you were supposed to be talking about that it's not going to hurt your grade, but don't talk about it in the presentation. Okay? So trying to get you to narrow your focus to the real nitty-gritty details of what should show up in the presentation. Make sense? And then I'll probably go through and actually grade them. So it'll initially just be narrowing the scope for your presentation. Or I might say the whole thing is perfect. Just present your paper. Make sense? The presentation will be group. Okay, so if you decided to work together on a topic, you will present that topic together as a single presentation. You don't need to do individual ones. Okay? Attendance is mandatory for the presentations because the theory, no one's going to want to play around with using the iPad to record because you haven't had experience with it. Okay, if you want to, you're more than welcome to, but I doubt anybody will. So you have to stay here, attend the presentations so that you can see what's going on within them. Okay? You will also grade the presentations. So I'll come up with a little grade sheet. Okay? And it's entirely on you on how you want to grade it. In that grade, I will compile everybody in the class, and that will make up 30% of the presentation grade for the people presenting. Okay? So your grade is in each other's hands, sort of. Okay? Which usually means everybody miraculously gets perfect scores. Okay? And then exam questions. From your presentation, I expect 5 multiple choice exam questions, which gets to our final. So there will be, at most, I think 15 presentations. 1, 2, 3, 4, 5, 6, 7. We got 13 people here. Probably 13 to 14 presentations if everybody did it individually, which would then mean 5 times that. We're looking at roughly 60 questions. I will pick the ones that I like the most. It's usually 1 to 2 per presentation. That goes into the final exam. So you should have access to that unless you present and just say, I'm not going to give you my questions, which is kind of mean to the rest of your classmates. But again, that's your choice. Kind of make sense? Questions about that larger assignment? Okay. to cycle through and find stuff. And I can't click between these. Maybe I can. So we had two papers. We had toward an atomistic description of urea denatured state of proteins, and then we go all the way down to the bottom. We had forces stabilizing proteins. Okay, so a lot of what we'll be doing today is just kind of talking about these and saying what you saw or did not see. So, any initial impressions? So you read it and you're like, oh, that's easy. Or did you read it and you're like, oh, that's really hard. Okay. Tell me more.
Protein foldings at least seem somewhat familiar since we just got done talking about it, but when the proteinuria one is just...
So this one was easier to read, the forces stabilizing proteins,
Yeah.
and the other one was like way over your head. Okay. So that's an interesting topic. Why might that have been the case? So I agree. I completely agree with that statement. I did pick this one because I thought this one, the one that's up top from our, what is this? We'll just call that Feb's letters. Okay. I figured that one would be easier to interpret and read than the other one. Okay. Which will... I am a bit of a child. This is probably one of my favorite journal titles. The Proceedings of the National Academy of Sciences. We'll just stick with that. So we have these two journals. Why is this one harder to read than the other one? Did anybody notice any sections that were potentially different between them? Do we see these sections immediately jumping out at us in this first one? Like, oh, that is a discussion. Okay, so there's a note. You're saying that somewhere in this, what? Don't do that. Let me scroll. Thank you. Somewhere in here, there's a method section. Oh, it was right there on the first page, and I can't point to it. But in this article, you notice that we had a method section. Okay. How do we know it's a method section? It says methods. What else about how it's formatted might call attention to it being different? Font size all of a sudden changed. Why would they change the font size? This gets into scientists being kind of jerks. So they can potentially cram in more information, but we could just make it longer. I mean, while scientists in general are on the greener side of things, they don't mind burning a few more kilobytes to build a larger paper. What is the point of a methods section? Explain what they did, how they got to those results, right? Okay. Why would we change the font? Okay, so the method section is going to be a lot of just technical information. Is our average reader really going to be concerned about the technical information? No, so let's make it smaller, let's get it kind of out of the way so we can immediately skip that section and get better to our content. Okay, so changing the font size can help in that respect. That's actually kind of being an optimist and why the font size is different. I'm always a pessimist. What is the point of peer-reviewed science? What does peer-reviewed mean? Reviewed. Other scientists need to review it. What are they doing when they review? Do they just read the content and say, yeah, that looks good? So they could critique it. What else? Repeating it. Okay? Being able to repeat someone's experiments is pretty useful. So we could go through and be like, all of your conclusions make absolute sense based on the data you've collected. But when I go through and run the experiment, I get different data. What does that mean about the validity of that article? It's not very useful. Okay? So we do want people to repeat our experiments. However, if they repeat them well in the first shot, what does that mean about your job security? Not very good. Because now somebody else can do exactly what you just did. So there's a couple things that come down to how we approach method sections. Number one, it needs to get written in such a way that somebody else can repeat it. Okay? So that's where you have to specify the equipment used, all of that. So we need that kind of repeatability within it. However, we don't want it to be so repeatable that we lose our job. Okay? So we do all sorts of stupid things to make it more difficult for somebody to repeat. Like, make it more difficult to read. It's a smaller font. Why do they not pick a bigger font? Okay? So we can change the font size. Okay? We'll also cram in tons of information in there. Okay? We'll use fancy words. We used a finite set of structures. How about we just used a set of structures? We used 20 structures. Okay? Their word choice is very kind of particular. They'll start using fancier words, and that happens in the rest of the text as well, because they're trying to sound smart, okay? Which they probably didn't have to try that hard to do. Okay? But it's also going to go through and make it very difficult to repeat. You need to be an expert to be able to repeat it. It's not just the average Joe that's going to be able to go through and do it, and even the expert, when they go through to do it, will probably screw it up the first couple times until they figure out there was that one key detail where they didn't include that when they were supposed to do the experiment, the color should have changed from yellow to blue, and that meant it was working. And that's all it was, but they decided to leave that detail out because that wasn't really necessary to repeating the experiment, but it was necessary to it being successful. Okay? So they're usually written very concisely, very straight and to the point, avoiding a lot of detail. Okay? So, back to the original question. We've now identified a method section because it says methods, font size is different. We now have an understanding of potentially why we'd make that font size different. Why does this have methods and the other one does not?
The other one just reviewing a bunch of articles.
This is an original research paper. What was the other one? It's a review of everybody else's research. Okay? It is still peer reviewed, but it's peer reviewed in the sense that someone goes through to say that you've collected all of these observations correctly. You're looking at a proper smattering of different scientific opinions, and you're then interpreting those opinions correctly. Okay? So it is still being peer reviewed, but it's not being peer reviewed to repeat the experiment. It's being peer reviewed to say your conclusions were drawn correctly. Okay? How do we know that this one was a review? So one option, it didn't have a method section. What else? Yeah. I didn't say it all had to be really hard. It says that it's a review. Okay? So if we think back to the assignment that I'm going to have you do at the end of the semester, which of these should you be trying to mimic? Probably the review. Which is nice, Mark, because you said this one was a hell of a lot easier to understand than the other one. Because the goal of this one is not to try and confuse. The goal of this one is to summarize all the results that are already out there so somebody else can go through and say, well, I think there's a hole here. I'm going to come up with research to fill that hole. Okay? So different goals in mind behind those parts. Okay? Or behind those actual articles. Okay? Other sections. How did we know we had a method section? It told us. How about an abstract? Okay. Both papers actually had an abstract, which is a bit odd. This one clearly states it, whereas the other one, and I don't have a freaking mouse. God, I really wanted these side by side. Irritating. Oh, Well, I can't move stuff. I can just go back and forth. Okay? This one does have an abstract as well. Come on, mouse. But it doesn't say abstract. Okay, well, what do you know about an abstract? What's its goal? Summarize the whole experiment. Okay? Where would you typically find an abstract then? At the very beginning. Both of these have an abstract because they both have a beginning. Kind of. That doesn't mean we have an abstract. Okay, Mark, you shook your head.
No, my computer sucks.
Oh, okay. I'm with you on that one.
Like I'm trying to do the same thing, getting them side by side, but it won't let me.
Oh, yeah. I'd like them side by side too. So how do we know this one has an abstract? What about this one has a characteristic? This is bolded. And if we go back and look at the other one, come on, it's also bolded. Okay? So it's trying to look for those visual cues that help pull us out and draw our attention to it. Why would we want the abstract pulled out and made more obvious? Catch our eye. Why do we want it to catch the eye?
Once we start reading that first, really kind of have
We want people to actually start reading it. Okay? The introduction is going to have a lot of background material for somebody that doesn't really know all of the detail behind what's happening, as I give you some more information about what an introduction does. The abstract isn't going to give you that information. What is it largely going to do? Outlines everything. Okay? So it will probably give us a goal. This one's really nice. The goal of this article, there it is, cleanly stated. Okay? And because I yelled at Jen all the time for this in labs, see, I'm not crazy. Sometimes we go blatantly out and we state exactly what we're trying to do. Okay? Whereas the other one, can I get back to the other one? They don't say the goal, but we present here. We're talking about what they're actually trying to do and put out into the world. Okay, so it's using the English language to clearly address those issues. Okay, here's an interesting thing. It says we. Do we encounter we very frequently? Not a whole lot. I mean, it does pop up every so often. Okay, but we is typically not accepted in a scientific article. Okay, it can float in there, but it's typically not. Okay? So, our abstract gets us our goal. Is that all it says? And Mike, you said results. Okay. Does our abstract give us some information about the results? Yeah. Okay. The idea is to summarize all parts of it. Okay, which brings up another part. We'll have results. Okay. So we've got the abstract, which gets us our goal. Guess what our introduction was trying to do? Explain the goal. Okay, provide the context that makes that goal make sense. Okay. We've got results. Okay, we do want to summarize the important results because the results were important enough for us to put in the paper. Okay, so our abstract, if it's supposed to summarize everything, should also include something about the results. What else should it include? Was there a goal for the paper? Yeah, there's a clear goal. There is a reason why the paper is getting written. Okay. What would you do at the end of a paper?
correlate results.
Probably correlate your results back to the original goal. Okay. So some kind of concluding statement that yes, this actually worked. The results presented herein are unique in providing a complete picture of the chemically unfolded state of proteins and contribute to deciphering blah, yada, yada, yada. That's exactly what the first thing says, except it now says, results did this. We did a good job. Okay? So it's trying to acknowledge the different parts of our paper and address that information. Kind of makes sense? What other sections are there? Conveniently stopped on another page that might be useful.
Introduction.
An introduction. Okay? What is the background behind why we're looking at this? Okay? How and why are these things important? Why should we be caring about any of this? Okay? Notice here they're also talking about a historical perspective. Why would we talk about the historical perspective in this paper in particular? This is the review article. We're trying to really provide the context by which all of the other things are going to be tying back to. If we go back to the original one that was a pain in the butt to read, okay, it has been known for decades, meaning the historical context. Okay? It's still bringing that information in. Even though it doesn't say introduction, it's providing the same information that the other one blatantly said at the beginning of its introduction. Does that make sense? You guys get enough of an idea of this that you're like, okay, Mike, stop talking about it. I think I can do this now. Yes? No? I'm seeing some head shakes, yes. So I'm going to kind of accept that.
All you want us to do in our paper?
In the one that
Look
you
at each of the sections, try and identify those sections, yes, and then say how they are different from everything else. Okay, what makes them stand out? What are they trying to do within that section? Okay, which in theory you've already done with many lab reports that you've written beforehand. Okay, but now it's kind of a self-reflection. You're reflecting back on all that stupid work we made you do in other chemistry labs. And now saying, well, what were those sections trying to do? Something that we really should have had you do at the beginning and not now at the end of your science career.
double spaced 1 to 2 pages?
Double spaced 1 to 2 pages, yeah. If you really want to run at it and you're like 5, 6, 7 pages, that's fine. I'm not going to penalize you for writing too much. I might not read it. We're going to be honest. If we're really going to be honest, I'll probably only read the first page. Okay? And as long as that seems like you've got the idea on what you're processing, what you were looking at, and how you talked about it, then I'll probably leave it alone. If that first page starts to look like you're summarizing, well, this one is talking about the intermolecular forces and how urea is interacting and how that changes, I'm immediately going to start reading everything. Because that's not the goal of the assignment. I don't care how urea interacts with anything. What I want you to do is to evaluate how they're presenting that information. How are you looking at this article and saying, this is distinctly different from that other article? Here's how they're different. This is a different section. I know this is different from the other sections because of how they're phrasing things. They're the same in both cases. The introduction is the same in both cases. They both started at the beginning of the paper. Okay? They both started talking about a historical context. One explicitly said historical context. The other one said in the past decades. Okay? Already bringing in a timeline. Does that make sense? Okay? In some of the sections, you'll be like, well, it's not in the other one. Right? If it's not in the other article, tell me why. Okay? Which was the methods section. Okay? Interestingly, in the review, we still have results. Why would we have results in the review?
Summarize the results from all the
They've got to summarize the results from somebody else's experiment. Okay? And in theory, they're also summarizing their conclusions, but then taking their conclusions as a separate experiment and saying those conclusions tie to these conclusions. Or those conclusions and these conclusions from a different experiment conflict with each other, which means we need to come up with a new theory to explain everything. Kind of make sense? What do you guys think? Is this a stupidly easy assignment that I should not be even bothered with, or is this actually kind of challenging, potentially interesting? You can tell me stupidly easy, that's fine. Or just challenging and I shouldn't have assigned it. Should we wait till after you write it? Okay. I don't really want to talk about it in more detail. I'm more than happy to, if we want to continue to go through and try and break these down. It's up to you guys.
I just have one more question. So do you want us to divide it up with like headers like this has? Like
Headers aren't a bad idea. Okay. Minimally, it should be paragraphs. Okay, and this is something I still find shocking. Is someone will go through and they'll write a report that is one paragraph. Guaranteed that was not one paragraph. Okay, so if you're going to change topics, give me a new paragraph. Okay, if you're changing topics that drastically, which you'll probably be doing as you shift from introduction to results, a header's not a bad idea. Yeah, I mean they did it.
Yeah.
Okay, they didn't. Okay, as long as there's a clear kind of difference, you don't need to provide a header, but it can be helpful for readers. Okay, and for those of you moving into future work, it can be helpful for anybody potentially grading your work. Okay, the less a grader has to work, usually the better your grade. Make sense?
And this is double space too.
Double spaced. Yeah. Write it up. Be like, I don't know if this is what Mike wants. Email. Bring to me in office hours. Hey, this is what I put together. I'll be like, yeah, that looks awesome. Where you wrote one paragraph, I think you need a couple more. Okay. At least pretend to make it to the second page. Okay. Make sense? Okay. With that, I'm going to attempt to make it to the PowerPoint slides. Well, let's just see what happens. I think that worked. Yeah, I think that actually worked. Cool. Okay, so I didn't entirely break my computer. Unlike Mark. Oh, so this was our scientific writing and interpretation, all that fun stuff, right? Oh, there we go. We'll meet in the library, February 23rd.
Did you post these slides or not yet?
Fair point, I have not posted them. You want me to post them right now?
I can wait.
I'll post them right now. No, I won't. Because I don't have a mouse. Okay, let's see if I can do this.
You don't have to do it now, I can get them later. You got pretty quick one.
When I have a mouse again.
Yeah. How did you break your house?
It's the mirroring aspect that I'm doing right now that's not allowing me to see my mouse. So I'm using actually the iPad now to move through this, which is kind of frustrating. But it's working. It's alive. I think that says it's uploading. I think it just said, it should be there. No. There. So, sorry about that. Should have gotten that up earlier. Okay. So we, I believe, ended last Tuesday looking at kind of our enzyme qualitative discussion. We mentioned the whole lock and key versus the induced fit, right? Can you guys summarize that for me? Which of those do you think is more real? Probably the induced fit, because I'll even accept a one-word answer on that. Forces. Right. We have intermolecular forces. When I bring two things together, those forces now change. If the forces change, what happens to the structure? The structure will likely change as well. Okay? So the induced fit is likely the better model. Okay? But there are some enzymes, even if they work under an induced fit system, when we study them, they equate to the lock and key. Okay? So just two pieces fit together and they made product. Okay? Nice and straightforward and simple. Okay? Some enzymes don't do that, which then sets us up with kind of a complex system of how we study these, and that is really the crux of Chapter 6 and Chapter 7, which is looking at those aspects, and it's a real royal pain in the butt. So if you thought the acid-base stuff was rough, this is fun. Right? So if we scaled this out, what happens, we had our enzyme and our substrate. They came together to form what's known as the enzyme-substrate complex, because they're now bound to each other. Right? Those then do something to form the product. Very typically, it's the enzyme still holding on to the product, and then the product is released. So we have these different stages within the reaction, which brings up the next point. If we have different stages or different reactions happening along the way, that means we'll have different thermodynamic energies. That means we'll have different rates, okay, for each of those steps. So if something were to come in and say, mess with any one of those states, how does that end up changing the overall result? And that's largely what's happening within biochem. We're looking at all of these reactions that are kind of spider networked out that all interrelate to each other. And if we mess with one of them, that's then going to mess with 4 or 5 others. We need to come up with some way to be able to predict and monitor regular changes so that when something changes, we know why and how it changed. Okay? And that is exceptionally challenging. Okay? But that is the goal of biochemistry. So, if we look at our enzyme, or this whole process, we've got the substrate and we've got our active site, and those are all interacting via our intermolecular forces. It's going to be a bit tedious to draw out these big pictures each and every single time, so what you'll end up seeing me do is abbreviate down to our standard symbols. E for enzyme, because enzyme begins with E. Okay? It's like a little Sesame Street. S is our substrate. ES is them now combined together. Okay? And notice that we have some reaction arrows there. What kind of reaction arrows are we showing between our reactant and our product? Equilibrium. Because in the process of running all of our reactions, we have the potential for the reverse thing to happen. I could have the substrate bind to the enzyme, and the enzyme just say, nah, I don't want it. And the substrate pieces out. Because there's no covalent interaction linking these things, okay, typically. So what we're looking at is just this weak intermolecular force. If there's enough energy associated within the system, the forces may very well break before we can do anything chemically to our substrate. Okay? So we will be looking at a forward reaction and a reverse reaction where nothing has happened. Okay? Is that going to make things a lot more complicated? Yes. This is why we have an entire semester of chemistry, 152, devoted to equilibrium reactions. Okay? And for those of you who aren't in 152, I know this is going to be a challenge. Okay? Bear with me, work through it as best you can. We do have parts where I'll just say, this is the end result, but this is how, I'm going to talk about how we got there. Okay? Which can be challenging, so just kind of work with me, but I will get to an end point and you can be like, okay, that's just what I need to memorize. So before we get too far into that, yeah, we can look at this thing known as PNP and what I like to call DADB. PNP is a purine nucleoside phosphorylase. So here's a little bit of nomenclature for you. What does that ACE signify? It's an enzyme. Okay, well, you steal all the thunder. Nothing new there. Okay, we have purine nucleoside phosphorylase. Okay, what do we think that enzyme is going to do?
Break down phosphorylase.
Okay, so break seems to make sense for enzymes, but do enzymes always break things? No, they can put things together. Okay, that's why we have catabolism and anabolism. Okay, so it could very well put something together. It could be breaking something. What is it potentially doing? Well, the phosphoryl gives us some information. The phosphoryl is saying it's acting on phosphate. Okay? So we are removing a phosphate. Okay? Are we removing a phosphate from just anything? What are we removing it from? Purine nucleosides. Okay, well, what is a purine nucleoside? genetic information, study that, and let me know what it is at the end of the semester. Okay? This happens to be a structure of a purine nucleoside. Okay? So what the phosphorylase will do is react with that and do something with a phosphate, which brings in an interesting thing. We might go through and say, well, it means that it's cleaving it. Okay? Interestingly enough, the nomenclature when we're looking at our enzymes usually comes from how it was discovered. So we could call it a phosphorylase and say, well, phosphorylases remove phosphates. And that's because when we took the enzyme out and studied it, what did it do? It removed a phosphate. Does that mean it couldn't do the opposite? No, it could very well do the opposite. What happens when we take that same enzyme and put it into a biological system? It could very well do the opposite reaction. So we have to be careful that the name just means, gets us some information about the type of reaction, but it's not telling us the direction necessarily. Because the instant we take the enzyme out of the biological system, we potentially change the activity of the enzyme, being it reacting in the forward direction or the reverse reaction, okay, which is exceptionally irritating when it comes to nomenclature. And it's not about to change anytime soon. Okay? So, what it will go through and do is metabolize adenosine into adenine. So its goal is to take the structure on the left and turn it into the structure on the right. Okay? Which is fun, because where's the phosphate? Not there. Okay, well, perhaps the phosphate is involved somewhere along in that reaction of what it's doing. Okay? But regardless, that's what it's trying to do. Which then brings up the next question, well, if this is what it's doing, maybe this is a good thing. If it's a good thing, I want to make it go faster. Okay? But too much of a good thing is bad. So maybe it's an enzyme that's overactive and I want to slow it down. This then comes in with this concept of drug targeting, which looks like a link, and I don't remember what that link is, so let's see what it does. It's not. That's good. Okay. Actually, I lied. It is. Yep. Oh, this is actually kind of pretty. So let's take a look at this. Let's pause the video for a moment. So what we're looking at that, or in that video, is just a way of modeling what's happening. And so our substrate slowly works its way onto the inside. Okay, well, if I want to inhibit this enzyme, what kind of drug might I pick? What would that drug look like?
Similar.
It should look similar to one of those two pieces, because what's happening is the protein's just kind of feeling around and says, oh, that looks kind of similar, I'm going to start to drag that in. And as it goes, oh, that's more and more similar, it drags it further and further and further into the enzyme to when it can get to the reactive site and it actually does the chemistry. So if our drug looks similar to it, okay, the drug can interact instead of our substrate. So if we look at possible substrates or possible drugs, we've got a variety of different structures here. And notice that a lot of them have commonalities with our original compound. A lot of them have dissimilarities. Because what are they focusing on? For instance, let's take a look at 4. What does 4 most match within our substrate? The OH part. Probably talking about that part. Okay? But if we looked at, say, well, they don't break it up nicely. Let's say 5+. What is that trying to mimic? The whole thing. Okay? So depending on which part we think is the most important for the protein to reach out and grab, we will try and mimic that. Well, how do we know it's the most important? Trial and error. Okay? So what someone went through and did is said, well, this is what it's targeting. I'm going to talk to an organic chemist and I say, here's the compound that I know my enzyme is targeting. Make me 20 things that look like this that aren't the same thing. The organic chemist, sure, why not? I'll go through and do that, okay, as long as you pay me to do it. Okay? They go through and make those 20 different things and they ship them out to the biochemist. And the biochemist then does what? Well, takes those 20 things and takes a 96 spot well plate or larger and drops a little bit of those chemicals into each one, and then drops the enzyme they're studying into each one, and then drops some kind of signaler that allows them to say, is the enzyme working or not? And then they look at the plate and say, well, that one stopped working, that one's still working, that one's kind of working. I know that one is the best looking structure. Okay? So now I go back to the biochemist and I say, well, I know I had you make 20 things, but this number 4, that seemed to work really, really well. So make me 20 more structures that look like structure 4. And they go out and they make 20 more structures and send them back to the biologist, and the biologist says, runs the exact same experiment and says, oh, we killed it some more. Okay? And then they say, okay, organic chemist, make another 20 that look like this one now. Okay? When do they stop? What's that?
Never.
No, that would be a bit annoying if we never stopped. I mean, we kind of never stopped. It's going to be a cost-benefit analysis. Okay? Because more than likely, it's never going to completely cancel out the activity. Okay? And usually, we don't want to completely cancel out the activity. Biology invented that enzyme for a reason. If we completely kill it, that could have a massive cascade effect into the rest of the system. Okay? So we'll want something that limits it, and then we have to decide how much does it get limited. Well, what's the physiological change that's happening further on down the line? So we're probably looking at an organic chemist, a biochemist, and a biologist, or even a clinical doctor. Okay? All working with that same kind of process, trying to decide how it best interacted and how that worked out for them. Because once we've gone through and designed that and say, yes, that kills that enzyme, let's give it to a person now. Oh, cool, it killed the enzyme. It also killed the person. Okay? Because it did this other thing that we didn't anticipate. Okay? So then we have to go back to our study and say, well, that one didn't work. Let's try that one. Give it another shot. Okay? So it's a long process that, as trial and error as it sounds, it is admittedly kind of legitimate science. Okay? We're making a hypothesis, we're testing that hypothesis, but our throughput in that making and testing is insanely fast because we've scaled it out to such a large size. Okay? So we can end up making much larger breakthroughs without actually understanding a lot of what's going on within it. Okay? And that's the part that I have an issue with. We should be intelligent enough that we can design something accurately enough that it works on the first try and does exactly what we want it to. The problem with that is we're looking at such a massive system with so many different interconnections that it's very difficult to nail that down perfectly. Right? And the best that we can come up with is kind of what I call a shotgun at the barn approach. Okay? You're blindfolded, you fire a shotgun, you heard a sound that you think hit the barn. So you turn and aim your shotgun more towards where that sound came from. Oh, you heard it a little bit louder. Okay, so we keep aiming, okay, until eventually we hit the target on the barn. Okay, it works. It's just a little messy, okay, and it could usually result in a lot of shotgun holes. Okay, so another example for an enzyme, we could look at chymotrypsin. So chymotrypsin, in a less kind of specific sense for this example, chymotrypsin catalyzes the cleavage of the carboxyl side of phenylalanine and tyrosine residues in a peptide. So we've got a long peptide chain. Chymotrypsin goes along, scans phenylalanine, cut. But it only cuts on the carboxyl side, not on the amino side. Okay? It'll do the same thing to tyrosine. Okay? Any guesses into characteristics of this enzyme? What does phenylalanine look like? Okay. Phenylalanine had that ring on it, right? What does tyrosine look like? What do you think might be in the enzyme pocket? What's that? Something that could interact with the aromatic ring, which would likely be another aromatic R group. Okay, because remember, our enzyme is composed of, more than likely, amino acids. So what we're looking for is something that would target and grab onto that and allow us to cut. There's an interesting other feature with this. It only cuts on the carboxyl side, not on the amine side. Okay, so what we have here is effectively scissors, but not just any scissors. They're like scissors that we use. If I gave you a pair of left-handed scissors, how many of you could use them? Oh, one person. Why is that? Because we're all right-handed. Scissors are chiral molecules. They have a certain handedness associated with them. Guess what proteins are? Chiral molecules. Remember, our amino acids were all L, okay? Or mostly all L amino acids, okay? Which means when we go through to cut a protein, it's going to have a handedness to it that will only allow me to get access to one side or the other, depending on the rest of the protein structure or the rest of the catalyst, our cutting catalyst. Okay? So if we took a look at this sequence, where would we cut? Okay, bless you. The dashes are looking at our chirality of the R group. That is the carbon to our R group. Is that what it says it cuts? It cuts the carboxyl side of our structure, meaning it's cutting the peptide bond on the carboxyl side of phenylalanine and tyrosine. So it can't be cutting the R group because that's not the carboxyl. So where's the carboxyl side for each of those? Okay, so the dash connects up. That's our carbon. Wow, eraser. Carbon. What else would we have? Oh, we've got the carboxyl side over here. What else do we have? The amine side there. Where's our enzyme going to cleave? Out there. Our carbonyl to whatever's out there. We don't know what's out there because I screwed that up. Okay. Will it touch any of these bonds for that amino acid? Why not? Are they phenylalanine? No. It can't see that enzyme. Where's the next one? Will it cut in here now or over here? We'll cut on the first arrow because that is our carboxyl side of our phenylalanine. It's okay. I didn't tell you to memorize them all. Okay. So we can look at our protein sequence and we can then start to cut based off of where they show up. Okay. So if we took a look at this protein and chymotrypsin was treated with it, what pieces would we have left over? Anybody see that, right? Cut this here. What was our one-letter code for tyrosine? I was pretty sure it was Y. I was going to be like, oh crap, I really screwed that one up. We got Y, and for phenylalanine? Okay, where's the carboxyl versus the amine side on our protein? Our carboxyl's on the far right. The far left is our amine side. So if we were going to go through and cut, where do we cut? Right of the Y and right of the F. Make sense? Okay. I thought this was neat enough to try and track down some pictures. So if we take a look at the active site of chymotrypsin, okay, we'll have, and this red thing in between or in the middle there is the binding or our target enzyme or target peptide that we're trying to cut. The enzyme has a sequence of multiple amino acids. Okay? Those amino acids are being used to help cleave and break that bond. Those amino acids being involved in the breakage, we have histidine and serine. Notice in the pocket for where our ring is, what do we have? We have glycine. There's no really aromatic structure there. Why would glycine be helpful? What was the structure of glycine? Hydrogen. Why is that helpful? What's that?
Proton donor.
Can't do hydrogen bonding. It can't be a proton donor because it's not bound to oxygen, nitrogen, or fluorine. Glycine happens to have, or be, really, really small. Phenylalanine, tyrosine, relatively big. The glycine being small can provide a little bit more space for that larger group to sit. Okay. Notice also that we aren't picking, well, it's kind of hard to even go much larger than that, but if we went any larger than that and looked at our polar one, what would be the issue with a polar amino acid there? It's going to potentially repel because we're looking at a hydrophobic ring. Okay? So it's looking at the different parts and pieces within those enzymes. I do not expect you to have those pockets memorized. What I want you to do is just get a feel for what's happening within the active site of your enzyme and how the different protein sequences can help kind of shape that pocket. Okay? And I think I've got another image here. Okay? This is actually showing the space-filling model of that chymotrypsin. Okay? The blue there being our phenylalanine of whatever peptide we're trying to cleave. The red and the green are referencing the individual, or sorry, it's not even showing phenylalanine, it's histidine. It's showing the amino acids that are directly involved in the catalytic breaking of the peptide bond. Okay? Remember, the peptide bond was super, super stable. Okay? So to break that, we're going to need a variety of different functionalities associated with that. We got serine with a nice hydrogen bonding ability. We've got histidine, okay, with that nucleophilic or negative nitrogen. We have aspartate, okay, our acidic or potentially basic hydrogen. So by shuffling the hydrogen around from different locations, we can change the functionality within the active site and help catalyze the breakage of that bond. Kind of neat. Okay. The next part of this, which I won't start talking about, I'm actually done, is looking at a kinetics review. Okay, so those of you that missed 152, that's what these slides are supposed to be doing, is trying to really kind of briefly whirlwind through how we discuss and look at the kinetics of an individual reaction. Okay? For those of you that had 152 recently, hopefully this is all review and you'll be like, yeah, that wasn't that big of a deal. Those of you who had it a long time ago or didn't have it at all, hopefully this will at least kind of narrow the focus and help you understand a little bit more what's happening within the context. Because I don't think the textbook does a very good job of providing that review. You'd really have to go back to a general chemistry textbook. Okay?